The overall goal of this procedure is to measure physiological activity of olfactory neurons in the Drosophila Antennal Lobe by optical imaging of intracellular calcium changes. This is accomplished by first assembling the mounting block, wire holder, and antennal shield. Next, a fly expressing the fluorescent calcium sensor GCamp is mounted and fixed to the block. Once the fly is mounted, the cuticle on the top of the fly head is cut away to expose the antennal lobes. The final step of the procedure is to set the fly under the microscope and stimulate it with an odor. Ultimately, results can be obtained that show odor-evoked activity patterns in the Drosophila antennal lobes through recorded changes in relative fluorescence of the calcium sensor, GCamp.
This method can help reveal how odors are represented as spatial and temporal patterns of neural activity at different levels of the olfactory circuitry.
The main advantage of this technique over other methods like electrophysiology is that we can record the activity of genetically identified neurons that are not accessible to recording electrodes.
Though this method can provide insights into the neural processing of odors, it can also be applied to other regions of the Drosophila brain, such as the auditory and gustatory systems.
First, thread the screws through the back of the plexiglass mounting block such that they do not extend beyond the front. Use a precision drill to make a dent in the upper border of the circular chamber for the fly by digging under the surface to reduce the thickness of the block. Under a dissecting scope, cut a copper grid with a scalpel perpendicular and close to the bottom of the slit to create a collar for holding the fly. Make sure that